and to give me this opportunity to talk to you about a method that we've developed that has kept me excited for almost 2 years now. And it's called thermal proteome profiling, and it enables tracking of drug action in living cells, but it's also really a novel way of measuring the state of a cell at any given time. A little bit of background, where I come from and what I work with. I work at the research company Celsom, which was started by EMBL in the year 2000, and we work with drug discovery. More precisely, we work with understanding the mechanisms of drugs. And I will start by outlining a couple of key problems in drug discovery that we and also many other people generally in drug discovery are trying to solve and address. And the key questions really in current day drug discovery, and they have been key questions for a lot of years now, are these. So first of all, there's two different branches of drug discovery. One is the so-called target-based drug discovery, and this is when you know from literature, from your own research, that a particular protein plays a key role in a disease, and you want to develop a compound, a drug that will inhibit this protein and hopefully cure the disease. What you do then is you develop a molecule which in vitro binds to that protein. But after you've done that, before you go into phase 1 or anything like that, what you really like to know is whether your potential drug actually enters a living cell and binds to the protein of interest, because this is not guaranteed by an in vitro assay. That's one problem. Second problem is you want to know what concentration of the drug you need to use in order to inhibit this protein if it does enter and bind to the protein in question. And the third, and also a really, really important problem, is the question, does my drug or potential drug bind to any other proteins in the cells? Because if it does, and this you don't know by just developing it in an in vitro assay against one protein, if it does that, it might cause very adverse effects and not be applicable for patients. So that's the target-based drug discovery. And the other branch is phenotypic drug discovery. In phenotypic drug discovery, you find potential drugs in a different way. Basically, you want to create a certain phenotype. A simple example of that is if you want to find a specific cancer drug against a certain type of cancer, you take cancer cells and you throw different molecules, different compounds on these cells. And you find the molecule that kills these cancer cells. And in this case, your phenotype is death. So it's a phenotypic screen, and the phenotype is death. And once you find this molecule, this could be a potential drug. But all the problems which are outlined above apply, plus one additional really big problem, and that is that you don't know the mechanism of the drug at this point. You know that it creates the phenotype that you are interested in, but you don't know by which means it does that. And so, why are developments needed in this field? So the current situation is such that for a large number of drugs, according to some people, for 40% of drugs that enter clinical trials, actually no evidence exists that they actually enter the cell and bind to the protein of interest. So many drugs fail just because of that. Drug off-targets, which are also a very common cause of a drug failing, are also rarely investigated. For instance, if you develop a kinase drug, you will test it against other kinases in an in vitro kinase panel, but that doesn't guarantee that the drug doesn't bind to any other enzymes in the cell, and I will show you examples of where this actually happens. And finally, for many drugs that come out of the phenotypic screen, which I just described, the target is not known. And then you have a molecule which elicits the effect you want, but you want to optimize it. You want to make it more efficient, less toxic, etc., but you don't know what to do with it because you don't know the mechanism, and it fails because of that. All of this requires new technologies, and we developed one such technology, thermal proteome profiling, which I will describe. And there are two parts to this technology. One is really genius biochemical idea by Bernd Nordlund, which came up in 2013, which I will describe. And then also recent advances in multiplex quantitative mass spectrometry, which were absolutely necessary for the development of this method. And I will start by describing the advances in multiplexed quantitative mass spectrometry. This is a typical proteomics workflow. You probably have seen that already, or something similar to that during this week, or you're familiar with it. You start with cells, with tissue, you extract the proteins, you digest them. Then what we do, we label peptides with isobaric tags and quantify afterwards. We use TMT, which I will describe in a second. We acquire the MS data and then we perform data analysis. So, how do we do quantification again? What is the concept of TMT? Basically, a naked TMT molecule is called naked because here the heavy isotopes are not positioned on this molecule, but this is what the naked molecule looks like. It has a reactive group which attaches itself to the N-termini and lysine side chains of peptides. So typically a triptych peptide will have either one or two such tags attached to it. It has a mass memorization group where different heavy isotopes are positioned in such a way as to balance the mass reporter group, which also has heavy isotopes positioned along it, so that all different tags have exactly the same mass. And the mass reporter group falls off upon fragmentation with, for instance, HCD, is detected in the MS spectrum and is used, this ion is used for quantification. And this is an example of two tags, the so-called 127 and 129 tags. It's called 127 because it appears at the MZ position of 127 Dalton in the MSM spectrum. And the way it works is that, again, you have a heavy carbon positioned here. You have the balancing heavy isotopes, including nitrogen here. HCD which occurs here. This guy is protonated, and the mass of it and the MOZ of it in the MSM spectrum is 127.13. For the 129 tag, it's a similar story, except that there are two additional carbons here, and consequently less balancing heavy isotopes here, so that in total these two tags have exactly the same mass, but the reporter ions will have different mass. That's the concept. And in total, from the beginning, there was 6 such different tags, encoding reporter ion masses starting from 126 going to 131. So it enables 6-plexing. And basically the workflow is as follows. You have your 6 different treatment conditions. You label them with isobaric tags, so TMT, 6 different conditions. They will appear as a single peptide ion in your MS spectrum because the tags are isobaric. But then upon fragmentation, the different report ions will fall off and create signals from which you can calculate fold changes and compare the different treatment conditions. In this case, everything was 1 to 1 to 1 to 1. It's a great technology. It's really super necessary in what we do and what other people do because you always want to compare different conditions. The problem is it comes at a cost. And when you label peptides with such tags, you create some side reactions, so the fragmentation gets a bit funny. It's a whole kind of topic in itself. The thing is that, as you might have already realized, that the more conditions you want to encode, the bigger you need to make this tag in order to be able to play around with the heavy isotopes. And the bigger the tag gets, the worse protein identification you get. As you can see here, so the protein coverage, if you have the unlabeled condition, if you go to 4-plexing using iTrack, that was the first isobaric technique, you lose a little bit with TMT, you lose a little bit more, and this is iTrack 8, with which you actually lose quite a lot. So we settled on TMT6 because that was a good compromise. We need high protein coverage as the most people, and we were not prepared to get high multiplexing, but lose protein coverage. And then there was a really lucky development a couple of years ago, we made this small discovery simultaneously with Steve Gige's group in Harvard and published the papers back to back. Basically, what happened is, it's a nice story because it was done for a different purpose and had a different result than expected. Basically, as I told you already, cleavage for this 127 and 129 tags occurs here. Now, there's another great fragmentation technique which you've already been told about, called electron transfer dissociation. It has a completely different mechanism to HCD. I will not go into that. But it cleaves here, okay? So it cleaves closer to the 6-membered ring, which means that you lose out on the coding that this heavy isotope provides, and you can't get as high multiplexing with ETD. And Tamer wanted to address that, so they created 2 new tags. again 127 and 129, where they simply moved this heavy isotope, or not moved, but instead of using 13C carbon, they used the 15N in this position, thus creating a new 127 tag, which had a slightly different mass, and the new 129 tag. This facilitated multiplexing with ETD. But what it also did, what we stumbled upon by kind of coincidence, was that actually with new instruments which have very high resolution capabilities, also in the MS spectra, you can actually begin to resolve the difference between these two different tags. So they have exactly the same nominal mass, but they differ a little bit here in the decimal. So 13108, 12476. And that's the difference between heavy and normal carbon and heavy and normal nitrogen, which is 6.32 millidalton. It's really minuscule, but nowadays when you acquire high resolution MSMs, which you can do very fast, the resolution of 30,000 at MZ 200 Dalton, you actually get bottom resolution of these two peaks, which means that you can now mix these two different tags, and you can expand your multiplexing capabilities from 6 to 8. And the great thing about that is that you do that without increasing the size of the molecule. The size of the molecule stays the same, and this doesn't affect protein coverage. This was really great and very enabling. And also later on, it became possible to use 10-plexing. So the next stage of the same concept was introduced. So same size molecule, but then different conditions. That was really good for us for many applications, but then there came one application for which this thing actually turned out to be absolutely essential, which I will describe now. And this is in order to describe the biochemical concept behind thermal protein profiling, I first need to describe the concept of a thermal shift assay. And the concept of thermal shift assay is actually something that has been known since 1959. It's a really long time ago, and that is the simple fact that if you have a ligand coupled to an enzyme, this enzyme will be more thermostable, meaning that an enzyme which is coupled to a ligand, if you heat it up, it will start to unfold at a higher temperature than if it is not coupled to a ligand. Very simple principle. And since 2001, this has been utilized actually in drug discovery in vitro assays. Because what people would do, they would take an enzyme without the drug, and so it's folded initially, then you start heating it up, and it begins to unfold. And you can read out the rate of unfolding by using fluorescent dye, basically, which will bind to the hydrophobic stretches of the protein which become exposed. If you do then the same experiment, but you add now a drug which binds to the enzyme, it will become more thermostable, and by the same readout, you can now establish that the melting point of your protein has shifted. It has become more thermostable, meaning that the drug bound to it. And this you can use for screening of compounds in an in vitro setting against proteins, and find basically compounds that bind to protein. If it binds to protein gets stabilized. If it doesn't bind, it doesn't get stabilized. It's a fairly simple concept. Now, a group at Karolinska in 2013, led by Per Nordlund and Daniel Molina, had a really brilliant, brave idea on how to do that in living cells. So what was that? So basically, if you have your typical normal protein, it will be folded. The hydrophobic residues will be buried inside the structure, and the hydrophilic residues will be sticking out. So it will be nice and happy and floating around in the cell. So you have the protein in the cell. Of course, you have all the other protein molecules in the cell. This is what it looks like. And now the idea was, okay, so if you now heat the cell to a certain temperature or to different temperatures, at certain temperatures, any protein will begin to unfold. So this is illustrated here by these straight lines. So basically the protein unfolds, and then the hydrophobic patches become exposed. And when they become exposed, they become sticky. So basically if these things meet together, they stick together. So on a very short time scale, aggregation happens in a pretty short time scale, these unfolded proteins will begin to stick together and form an aggregate. Okay? The unfolded protein will still be perfectly soluble, but these guys will have built an aggregate. And what you can do next, you can now perform cell extraction, and then simply spin down your sample. And then the soluble proteins will remain in the supernatant, but the aggregated proteins will fall down and will become part of the pellet. And then by carefully extracting the supernatant and measuring the protein content at different heating temperatures, you could then infer what is the melting behavior of a specific protein. That was a very brave idea. Why is it brave? Because I think before people realized this idea, other people would have said that this is just crazy because this is what the inside of a cell is supposed to look like. It's extremely crowded. It was just a simply very cool idea, and we need more ideas of this type. These guys at Karolinska did that, and it actually worked. So exactly this approach worked. And they published that in Science in 2013, they tested it on a handful of drug-protein combinations. For instance, they did it for the drug called methotrexate, which inhibits the HFR, and very clearly when the drug was added to the living cells, the protein would become stabilized and melt at a later stage, melt and aggregate at a later stage compared to the untreated condition. When we read this paper, we were super excited because that seemed to us a perfect combination with multiplexed quantitative proteomics. We were quite full of disbelief initially whether this would actually work. But we set out implementing this method, and as you can already tell, this 10-plexed approach is really key for doing that because you want to have as many points along the melting curve as possible, and you don't want to do too many experiments. So this is the setup of the approach that we developed. You start with treated or untreated cells, or you also start with treated or untreated lysate. Both approaches work. And sometimes it makes sense to do both, as I will explain later. Then you take 10 aliquots of the untreated and treated condition. You heat them to different temperatures, starting from 37 going up to 67. So you heat for roughly 3 minutes each. You heat each aliquot for 3 minutes. Then you perform cell extraction, and you label the untreated sample with 10 team T-tags and the treated sample with 10 team T-tags. So you get 2 experiments in the end, so you need to perform 2 different mass spec experiments. And in this mass spec experiment, you then get a quantitative readout for the vehicle condition and for the compound condition. And this is what it will look like for many peptides. This is basically represents this melting curve, which looks like this. And then you can compare the melting behavior of the different proteins and fit melting curves, and then determine whether there's a significant shift between these melting curves. We had to develop software for all of this because it didn't exist, and this will also become publicly available pretty soon. And this here is the first picture of a melting or aggregating cell, whichever way you like to call it. So in this case, we identified roughly 5,500 proteins in K562 cells, and we mapped out their melting behavior. And as you can see, most proteins have pretty differential melting behavior. Some melt really early, blue means that the melting curve goes down. Some melt late. Some show this really funny behavior. It looked like they were going up. And that's a very important point, which I'll come back to at the end. All these initial experiments were done without detergent, for fear that if you would use detergent, that you would resolubilize the aggregates. And the aggregates you do not want to resolubilize because they're essential for your readout for this technology. So what happens to these proteins, which look funny, they're actually part of organelles. So if you don't use detergent, you don't get proteins out of organelles, but when you heat them to a sufficiently high temperature, the organelles start to break up and the proteins leak out into the cytoplasm. But for the majority of cytosolic proteins, actually, we got really nice melting curves, roughly 75 to 80%. And then we did some fundamental analysis, first looking at reproducibility of melting points of different proteins when you perform the experiments on cells, and as you can see there's really good agreement. And then also the melting point reproducibility when cell extracts were heated and not cells. Again, you see very good reproducibility. Funnily enough, when you compare the melting behavior between intact cells, when you heat intact cells, compared to heating cell extracts, the agreement was quite poor, so there was a very big difference. And there are multiple reasons for that, and we figured out some of them. I'll just tell you about one. And that is when you break up a cell, so when you perform cell extraction, you very strongly dilute the natural cofactors occurring in the cell. So for instance, the concentration of ATP will be much lower in cell extract than in the cell, which means that many of the ATP binding proteins will not be coupled to ATP, which then might mean that they become destabilized, which is exactly what you observe. Basically, in a cellular situation, ATP binding proteins melt exactly or even slightly more stable than all the other proteins, but in cell extract they all melt significantly earlier than the other proteins. The reviewers later asked us to also perform a dedicated experiment to really prove that, which we did by basically treating cell extracts with a high concentration of magnesium ATP, and we did exactly observe that the population of ATP binding proteins got stabilized. So that is one of the reasons why there's a difference here. So it's a fun fundamental thing. What we were really interested in was the drug case. So we treated initially cell extracts with diasporin and performed 2 biological replicates, and very nicely we were able to see that a lot of proteins had significant shifts in their melting behavior between the treated and untreated condition. The proteins that were detected overlapped very nicely with the known clients of diasporin. Diasporin is very known, very well studied pan-kinase inhibitor, so it inhibits a lot of kinases. These are some examples of the melting curves. So for instance, roughly 90% of the proteins got stabilized, a few proteins got destabilized. There were also cases, and it's important to keep in mind, where a known client did not show a melting point shift. It's important to keep in mind, so I would estimate that this approach works for 70% of the protein or so, based on all the experiments we've done. For 30% of the proteins, you will just not observe the shift. This is the example of SLK. Here, there's actually a pretty good explanation, because SLK is known to only bind diasporin when it is activated, and it is known that only a couple percent of it are activated in a living cell. So even if the activated form might shift, you don't see it against the background of the unactivated form. There might be other examples where proteins have 2 domains, and you inhibit this domain, but this domain just always melts earlier than the other domains. These proteins will just not be tractable. Okay. Once you find the targets of your drug, the next thing that you want to do is to establish the potency of your drug on these targets, because that's a very important parameter in drug discovery. And for that you use a slightly different approach to basically turn the experiment on its head. So for instance, if you know that for one protein you have a stabilization which looks like this, and the melting curve behavior like this, and upon drug treatment it gets stabilized like this, then you perform an experiment at a fixed temperature. So you heat all your samples to 53 degrees, and you treat the different samples, or the different, and treat the cells with different concentrations of a drug. And basically the concept is then that if you have a vehicle condition, you will basically measure this amount of protein. If you increase the concentration of a drug maybe here, you will end up here. And then when you have a high concentration of the drug, you end up here, and then after that you saturate. And this enables you also to determine at which point was 50% of the stabilization effect achieved with the drug. And then you hope that this correlates actually with the potency of drug binding to the protein. Again, this is the schematic for this approach. It's fairly simple. You treat cells or cell extracts with different concentration of a drug. You perform, in this case, one experiment, but you should still do biological replicates. And then you analyze the data, quantify the proteins, and if the protein was not affected, it shows a flat response. If it was stabilized, it will show a dose-dependent response. If it was destabilized, it will show a dose-dependent response like this. These experiments are highly reproducible. When you do two biological replicates, this is what the reproducibility looks like. So actually the quality is really, really good. And when you compare it to completely different assay for measuring binding affinities, this is a method called kinebits, which I will not go into, which is developed specifically for kinases, you actually also see a very good agreement. This is a very good agreement in drug discovery terms when comparing two completely different assays. Coming back to the staurosporin example, as I told you, we found many clients of staurosporin which were known, but we also found a few clients which were not known, which were actually not kinases. So coming back to the thing I talked about in the beginning, that you can have off-targets outside the target class. And we found actually several proteins that were affected, which are part of the heme biosynthesis pathway. In particular, ferrochilitase was affected by staurosporin. And ferrochilitase is a very interesting protein because it is responsible for the final step of heme synthesis. It basically inserts the iron into the heme molecule. And if that step doesn't happen, you have problems because this molecule is fluorescent. So if you have an excess of these molecules in your body and you go out into the sun, they will begin to vibrate and it will hurt a lot and you will get burnt. And it is actually known that photosensitivity is a major side effect of some kinase inhibitors. So we figured, could this explain the photosensitivity of some kinase inhibitors? So we did a small study where we basically took 3 marketed drugs, either marketed or phase 3 drugs, 2 of which, alectinib and vemurafenib, are known to cause photosensitivity, severe photosensitivity, and 1 which is known not to cause photosensitivity. And sure enough, it turned out that alectinib and vemurafenib inhibited ferrochilitase, while crizotinib didn't, making this a fairly plausible hypothesis why these drugs cause photosensitivity. Once you know that, it's actually, in terms of drug discovery, very easy to avoid these side effects for new compounds. You just add an additional screen against ferrochilitase. We also extended this approach to different drug classes, so also to epigenetic drug classes. For instance, we used the cancer drug panabinostat, which inhibits histone deacetylases. And here we actually got 100% results. So in terms of all the known targets of panabinostat that we identified, showed a nice thermal shift. And actually, when we determined the potency, they also had the potency that you would expect from panabinostat on cells. Also interestingly, we found a completely unknown target of panabinostat, TTC38, which we have since then actually proven is a direct target of panabinostat, but we still don't know what is actually the biological relevance of this target. But the potency of panabinostat on this target was as high as on its primary target. And this is part of accepted Nature protocol articles, which will also make all the software available for this approach. Okay. And then one more thing, what we found out and what was not known before also is that you can also use this approach to detect indirect targets of drugs. So basically when you treat cells with a drug and inhibit the protein, you will of course affect the downstream pathway. And it turns out that the proteins which are part of this downstream pathway will also change their melting behavior, which we observed with the marketed drug dasatinib, which introduced a significant shift in the CRKL protein and also SHP2 protein, which are both part of the BCR-ABL pathway. And in order to prove that, so the data was very solid for the on-cell experiment, but in order to prove that you have an indirect target without this a priori knowledge, you actually need to perform the in-lysit experiment, because in lysit you of course disrupt all the signaling. So if you have an indirect target, this target should not shift in lysit, which was exactly the case. So in the cell extract situation, CRKL shows no shift, which proves that it is an indirect target. And also to further prove that you can actually detect key differences between different cells, this also holds true when comparing K562 cells, where the BCR-ABL pathway is constitutively activated, so it's always on, and Crico is always phosphorylated actually in this case. Comparing this case to sugarcane cells, where this pathway is simply not on. And you see exactly the same difference as you see in K562 cells with and without drug treatment. So that was great, it was very exciting. This approach was actually voted as one of signaling breakthrough of 2014 in the Science Signaling magazine. But there's one point, and that is coming back to the fact that we use PBS for extraction, which is isotonic, it's not a detergent. So we're not actually solubilizing membrane proteins. And the targets of many drugs are membrane proteins. So we really wanted to know whether we could identify membrane targets. And for that, you need to use detergent. And we tried this approach with a mild detergent, NP40, which very nicely extracts membrane proteins, but it's a mild, non-ionic detergent. And I wanted to find out whether this approach still works then. And turned out that it does. So if you now look at the protein melting behavior in cell extracts, the melting behavior of the protein actually changes. They melt earlier with detergent. But in general, when you compare the melting points shifts introduced in this case by treatment with ATP, you see that the melting point shifts remain. So even though you use detergent, you don't resolubilize the aggregates, you still get your readout, and the shifts remain. But now comes the important question, what happens to membrane proteins, which you now identify? Do they shift? And turned out that they do. So these are two proteins which are transmembrane proteins, which are not at all even identified when you use PBS, but they are nicely identified when using NP40, and they're actually ATP binding, and they're indeed stabilized by ATP. So we're very excited about this result. And we also did the same experiment, same type of experiment on cell, this time stimulating cells with pervanadate. And again using PBS extraction in one case, in the other case PBS with addition of NP40. And in this case, because of course you heat the cells, aggregation happens inside the cells, then you perform cell extraction with the mild detergent. Since we already knew that the aggregates would not be dissolved, we would expect that the proteins would not change their melting behavior, which was exactly the case. So the melting behavior of proteins stays the same, regardless of whether it's NP40 or not. You get a lot of membrane proteins, as you typically get with NP40. Their melting behavior on average is slightly more stable than all the other proteins, but again, it's pretty heterogeneous. All the shifts that you get with only PBS, you also get with NP40. But again, very exciting, you also shift membrane proteins. In this case, CD45, which is the main target of pervanadate. It's covalently modified by pervanadate, and this is another protein which is a known transmembrane protein in insulin signaling. So this is the latest, most exciting news in this method. An additional thing, I already told you and showed you that you can get indirect targets, and I had this example with CRKL and SHIP2, but we also wanted to see whether this applies on a broader scale. And pervanadate was a perfect example of that, because it triggers a lot of signaling inside the cell, in particular activating the T-cell pathway, T-cell receptor pathway. So what we did, we took all the proteins which had significant shift and threw them in the Ingenuity engine with the background of all proteins identified, and the T-cell receptor pathway actually really nicely lit up, which shows that this approach is really applicable for tracking downstream events of drug treatment as well. So this concludes the talk. I hope I've shown you that this new methodology enables the monitoring of drug action in living cells and extracts and also tissues, in fact. You can potentially find markers for barrier signaling in cancer, for instance, because you can detect downstream events. It works for membrane proteins with mild detergents, and in general, it's just a new way of assessing the state of the proteome, which we're all very excited about. So with that, I'd like to acknowledge some key people. Pretty much everyone at Celsin for creating really wonderful infrastructure which makes these projects possible. In particular, these people who contributed directly to this study in an innovative way. Bernhard Küster, our favorite collaborator, Per Nordlund at Karolinska, and Wolfgang Huber and the joint postdoc Dorothy Charles at EMBL, with whom we just now got this SNACE protocols manuscript accepted, which will make all the software available and also describe all the experimental steps for performing these experiments. And I would like to thank you for your attention. Thank you.